couple of years. There we go. Recording in progress on a project that I won't talk about today about retinal metabolism, but I would love if you ask questions about that. We're doing some really exciting work, and you'll be able to tell, hopefully, in the introduction of my talk, why Tudor and I see eye to eye so well and things about the way that cell types work and our ideas about the next steps in neuroscience, because he just summarized it so beautifully about why I think the retina is the place where we need to do a lot of these things. So yeah, it's really an honor to be here, and I hope to introduce some of these concepts to this broader audience. So, the title of my talk, like Tudor said, is The Humbling Complexity of Retinal Circuits. So, I'm going to talk about multimodal classification of both amacrine and ganglion cells and how they help us understand visual processing and really neuronal processing and circuits more generally. So, before I get into that talk, I just want to introduce kind of the breadth of what we do in my lab now. And, feel free to ask questions about any of this stuff because I could talk for hours and we don't have that much time to talk, but I will talk about some pieces of this. My lab has expanded in the last few years, and we study the neurobiology and computations of the early visual system in mice at a variety of levels. So, sort of on the lowest level, we study neuronal biophysics, and I'll show you some of that today. We think about things like the way dendrites integrate, the way axons work, the different sodium channels in different cells. We work on retinal circuits and discover who's connected to whom in the retina and what the types are. We do a lot on retinal ganglion cell types, on amacrine cell types that I will show you today. Increasingly, I'm interested in their brain projections. There are 40 some odd retinal ganglion cells that I'll show you today. They actually project to more than 50 locations in the brain, not just LGN and SC and the ones you've typically heard of. So we're trying to solve that problem, figure out where all the ganglion cells project. We've done some work on behavior that's going to come out soon, on social behavior. And I'm working with engineers now on retina-inspired camera sensors for autonomous vehicles to use some of the computations that we've learned in the retina and put them on actual cameras. Additionally, I have some translational interests in diabetic retinopathy, in ganglion cell glucose metabolism, which has to do with lots of different retinal diseases potentially. And that's the project that Tudor and I are working on together. And I also have some interest in myopia. So today I want to focus on the question of what are the parts of the retina? How far do we need to zoom in to understand visual processing? This is, of course, a question that could be asked in any other part of the brain as well, or the central nervous system. What are the computational units? Is another way to say this question. How far do we need to zoom in to understand? We need to get to the protein level, to the brain area level, to the circuit level, somewhere in between. So, of course, there are classes of cells in the retina. Before we talk about types, people often talk about the five main classes: photoreceptors, horizontal cells, bipolars, amacrine cells, and ganglion cells. This has been known for 40 or 50 years now that those are the five classes. But within each class, of course, there are individual types. And for some of these classes, we know the types well. For other classes, we're still figuring out the types. But I would argue, and I will argue in a few slides, that our knowledge of the cell types in the retina exceeds that in most other parts of the central nervous system. But then the question comes down to, if you understand the type, are all the individuals within the type the same? Do we have to start to really look at individual differences in cells and their plasticity or anything like that? And then in the last part, in the last part of the talk today, I'm going to talk about the other elephant in the room, which is that what if the cells themselves are not the computational units? What if there's subcellular processing in subcompartments and synapses? Maybe we can't stop at the level of a cell. Maybe sometimes we have to go down to smaller components of cells to find out where the individual computations are happening. So, The retina, this is so that everything I just introduced could be applied to anywhere else in the central nervous system, but the retina is really in a position to lead the way because there's some unique advantages among vertebrate circuits in the central nervous system. I sometimes talk to my colleagues in the invertebrate world, the fly folks, and it's like the retina of a mammal, especially the mouse, where we know the most, lies at the midpoint between our understanding of the fly nervous system, where we have a name for every cell and we now have the full connectome of the whole animal, and the mammalian everything else, the cortex, the everything else in a mouse, where we know brain areas, we know some genetically identified types, but we're not at the level of knowing all the types and where they're all connected. The retina is kind of in between those two. So, why does it have some unique advantages? First of all, it's an ex vivo preparation that I'll show you in a moment, my ex vivo preparation, and that many people in the field. That means we call it ex vivo because it's not a brain slice. We're taking the entire retina out of the animal and we're using its natural stimuli. We're not using electrical stimulation or zapping some axons from somewhere. We're using light, which is the natural stimulus. And we are getting the natural output, which is the spikes that go down the optic nerve. We also have large EM reconstructions available. This was the first place where connectomics was done at scale. And the serial block face EM was done first in the retina in this 2013 paper. We have museum.iwire.org that my collaborator Sebastian Song has made, which is really an amazing website where you can go explore the morphology of the 47 morphologically identified ganglion cell types and look at their mosaic. So we have these dense reconstructions where the entire thing is traced and you have every cell traced. But finally, one of the most important advantages isn't a technical advantage. It's actually an inherent advantage of the biology, which is that retinal cells form mosaics. So what I mean by a mosaic, for those you who don't know, is that the cells tile in a noisy but still there hexagonal grid. And that's a organizing principle of the retina because you need to see everything everywhere. You don't want to have blind spots in your visual field where some cell types are represented and others are not. So that means that you have this self-repeating structure of cell types, and it's an independent check that you got it right. So this was one of the earliest papers on the left there in 1981 that showed the mosaics of dendritic fields. And it's also true functionally in receptive fields. And when you draw these contours around the receptive fields, you get this perfect mesh mosaic that everything meets. And that is an independent check that you got the cell typology right. So while the retina is in a position to lead, it also has been kind of cast in the wrong light, I would argue, in the literature. And that's because it's not a camera. The output of the retina, the streams that go out of it, are not a set of pixels. And of course, as neuroscientists, you don't really think it's a set of pixels, the people in this room, but I'd argue that even in textbooks and even what we're teaching graduate students, it's kind of still captured as a glorified set of pixels. They're basically center surround, right? They're Gaussian receptive fields, which is like a pixel with a filter on it, two-dimensional filter and some temporal filtering, but that's still fundamentally transmitting a signal about light intensity at any particular location or group of locations. I'd argue that a better analogy is a prism. So spatiotemporal patterns of light come into the eye, that's the input, but the output is not one thing. The output is not one set of filtered pixels like in a video camera. It is a wide array of features, all transmitted in parallel to 50 different places in the brain. So 7 or 8 colors in that rainbow that I have from the Dark Side of the Moon album, but it's actually more like 40 different output streams that come out of the retina. So let me draw it another way. So the optic nerve, it's all these different colors because there are 40 different ganglion cell types. And at any moment, spikes are going down the optic nerve. Let's talk about the meaning of those spikes. Those spikes do not mean that there was a bright pixel of light at this location. That's what the bits in a video camera mean when they go down the wire. The spikes in the optic nerve are about features. That spike literally means a bar was moving left to right around this location. There was optic flow here. There was orientation at 90 degrees in this general region. Something moved in this speed range here. And many, many more that I don't know because we haven't figured out exactly what the feature selectivity of each retinal ganglion cell is. But there are 40 features at every location all the time. So any location in the visual field sends 40 parallel messages to 50 locations in the brain about features of what's happening in the visual scene. So it is a qualitatively different kind of code than a pixel code. And this is what I'm not going to talk about my work with the engineers today, but this is what I'm trying to replicate with camera designers, because my argument is that this is the only way that autonomous vehicles are actually going to work in the future. Think about a video camera on a Tesla or on a drone or something like that. If you do the math, you can have a high resolution camera as much as you want. The electrons don't move fast enough down the wire for you to actually fly at 100 miles an hour or do fast movements. Like it doesn't even matter how much post-processing you have. The electrons don't move fast enough for a megapixel camera to actually transmit enough information fast enough to do anything. Yet we have birds flying around with much slower transduction in their eye, right? Biological transduction, not digital transduction. And these falcons are flying around at 200 miles an hour and not hitting stuff. That's because that only works because their optic nerve works like this. It transmits features, not pixels. And the motion processing and all is done in analog circuits within the retina. So that was an introduction to sort of my view of this. But this is why we need to look at the parts. If we're really going to understand how this works and how this feature selectivity works, we need to first understand the parts like we do for any computer system. You have to understand the difference between transistors and resistors if you're going to understand how a computer works. The same is true of understanding any neural circuit. So I'm going to tell you about some of our work on the parts list of the mouse retina in 3 different acts today. One is going to be on the retinal ganglion cells. As Tudor introduced, my lab's done a lot of work on this. I will try to keep this part relatively short because it's published, but I'll take you through the methods a bit and what we did on the retinal ganglion cells. Then I'm going to talk about the displaced amacrine cells, which is not published at all, with somewhat similar techniques, but a few differences as well. And then finally, I'll address the elephant in the room about the functional units, right? I'll tell you what the types of ganglion cells and the types of amacrine cells are, but then we'll come back to the issue of whether the computational units are even the cells themselves, or are they sometimes parts of the cells? Okay, so first, this work on the ganglion cells was led by a former postdoc in my lab, Jillian Getz, and also co-led by Zach Jessen, who's now a postdoc in the lab, but many others contributed. Most people in my lab, this was a 8-year, 9-year project. So many of the people in my lab over the years, we collaborated with Josh Sains at Harvard, Jeremy Siegel at Chicago, Karthik Shekhar at Berkeley, and others. So how did we define cell types? Again, this slide could be about anywhere else in the brain as well. What defines a cell type? I'd argue there's these 3 pillars. There's the function, the morphology, and the gene expression. So the function of a cell is what stimulus does it respond to? What's its feature selectivity? The morphology is what is its shape and its dendritic and axonal morphology. And the gene expression is which genes are turned on. And importantly, none of these pillars by themselves is sufficient. That's why I kind of like to think of it as like a three-legged stool, right? You'll see there's hundreds of papers that try to define every type of cell in a brain area by one of these criteria. One is not enough. Two isn't really enough to hold up a stool either. It's better than one, but it's not, right. You really need all three. So just a transcriptomic profile of all the cells in an area, you can cluster it into as many types as you want. Those are clusters. Those are not necessarily cell types. But when you have information about function and morphology and gene expression, you have a much better chance at getting the cell types right. So in the retina, this is what this looks like. The function is the light responses of the cells. It's the natural function. The morphology is the dendritic morphology of the cell. I mean, the projection patterns in the brain are also important, but we can't measure that in the ex vivo retina yet. I'm working on that problem separately. So it's dendritic morphology, and then it's gene expression you can do with single cell RNA-seq. So when we were working on this project, of course, we weren't the first group to think about what are the cell types of the mouse retina, what are the ganglion cell types. A lot of really high profile work on this problem had been done and kind of culminated in these three main papers that looked at each of these modalities independently. So in function, there was this Nature paper by the Bodden and Euler group that found 34 plus functional groups from calcium imaging. So there's two photon calcium imaging of the retina, and they just present a lot of visual stimuli, a battery of stimuli, and they do sparse PCA clustering. That's really this whole paper. There's the morphology work that I talked about from Sebastian Sung's group. That was the dense EM reconstruction of an entire section of mouse retina. So that was nice in the sense that it was unbiased. They traced 381 cells, and those were the 381 ganglion cells that were there. So they traced every one of them, and they tried to fit them into morphological types, including mosaic information. So I considered that pretty nice ground truth, with some exceptions being that it's still a small volume. So the big cells were cut off and had very few examples. And then there was the gene expression, which was done originally in 2019 by the Sains lab, where they just dissociated retinal ganglion cells, used 36,000 of them, and clustered them with machine learning into 45 clusters. So at this point in the field, the number of ganglion cells, we were starting to get some agreement that it was around 40, right? These three papers had reasonably similar numbers of types, but here was the big problem. The actual correspondences between the function and the morphology and the gene expression were few and far between. There was a couple where we had a real standout marker gene where we knew that that gene must mean that it's this morphology in this cell, but nowhere near 40. We had like 5 of those. So, my lab tried to fill this gap by making multiple of these measurements from the same cells. So the way we do this is we dark adapt a mouse overnight, dissect its eye with night vision goggles so we can keep things in complete darkness, flatten the retina, the whole retina out on a glass cover slide and show it movies with a digital projector while making cell attached or whole cell recordings with electrodes, right? So we can target cells in fluorescent lines, or most of this work was done just in wild type animals where nothing is fluorescent. We can measure the function of the cells, of course, by recording their spikes and their light responses. We can use whole cell and fill them with a fluorescent dye and reconstruct their morphology. And then we can also take another pipette, suck out the soma completely, and do patch-seq, essentially. Grab the soma and do single-cell RNA-seq on it. So all these methods are possible in the very same cells. So, long story of many years in recordings cut short to one slide. We recorded almost 2,000 cells. There's probably 3 or 4,000 cells in my database now. And we clustered them by their function first. But then for a subset of them, we also filled them and had the morphology data so we could align it with the morphology data from Sebastian Sung's data. And for a different subset of them, we also did the single cell RNA-seq. So working with Josh Sains and Karthik, we could align those cells to the gene expression. And then the most important thing, in my opinion, about this was that 2 years before this was published, we, during the pandemic, made a website to distribute all of it ahead of time, and that's called rgctypes.org. So here's a screenshot from that. You can expand this table and look at all of the 42 types we found, compare the light microscopy images we have to the EM images, look at examples of their light responses, see how they respond to spots of different sizes, look at the density of where they all are in the retina, and you can download all of this data and play with it yourself. So we made a web portal for this. So feel free to go check that out. So one of the key questions that reviewers had in this paper and that was nagging at me all this time is recorded 2,000 cells. You'd think I'd hit some of most types at that point, and we should be nearly complete in our typology, but how nearly? Like how close to 100% complete are we? And that's a very hard question. There's no like perfect way to answer that question, but I thought one way that would be a challenging, fun exercise to try to get there would be to simply record every ganglion cell in a region and figure out how many of them we could classify. So that's what we did. And this is a picture from the supplement to that paper where I was doing this recording. We ripped a hole, very carefully ripped a hole in the inner limiting membrane and recorded every cell, trying not to kill any of them, and figured out how many of them we could put into one of our types. And then post hoc put a marker for all ganglion cells on there. So we would know if something was a ganglion cell, but we couldn't get it to respond or we couldn't classify it. And the answer we came up with was 89%. So pretty good. Like we're not done. There are some types, a few of them that I call my white whale. You can look at the iWire database. There are a couple dense types that are clearly I should have recorded hundreds of times and never found, which to me means that my battery of visual stimuli were not sufficient to make the thing spike. So that probably is the case for a few types. So I think the real number of types, we found 42. The real number is probably between 45 and 47 or something, but that's pretty good. We're 90% of the way there essentially. And this is something that I think can't be said in most of the rest of the central nervous system, that we're anywhere near 90% to knowing what all the cell types are. So to summarize this part on the ganglion cell classification, we've identified 42 types. That's about 90% of the ganglion cells that are accounted for. The functional types, I didn't show you all the data on this, but the functional types align beautifully to the morphological types in the iWire Museum, really one-to-one, with it being very clear which type is which. The only exceptions are the ones that are poorly reconstructed in the morphology in the iWire Museum because they're huge and they're cut off. That data set was only 300 by 300 microns, and some of the cells are considerably larger than that. As far as the transcriptome, some of these morphofunctional types align very well with transcriptomic clusters. Others don't so well. So there were 45 transcriptomic clusters. We matched about 25 of them. Part of that's because we don't have enough data. We should just get more FuncSeq experiments. They're expensive. But part of it also may be that something that looks like a nice cluster in UMAP is not necessarily a cell type. So that's something you should always like look at those transcriptomic maps with a grain of salt. You know, you're going from 8,000 dimensional space to 2D space in UMAP and making things cluster. Of course, it's going to look like clusters, but that doesn't mean that those are necessarily cell types. Plus, a huge amount of the transcriptome depends on conditions and time of day and circadian and all sorts of other stuff. So It's just because it's a transcriptomic cluster doesn't mean it's a cell type necessarily. Okay, so let's move on to the amacrine cell work, which is currently in progress. So this is being led by a postdoc, really talented postdoc, Yulia Fedjukov in my lab. Again, a lot of people have contributed as well. So what are displaced amacrine cells? So this is a busy slide. Let me take you through it. So the ganglion cell layer at the bottom of the retina is so called because the vast majority of the ganglion cells live there. But actually, in a mouse, 2/3 of the cells that live in the ganglion cell layer are not ganglion cells. They're displaced amacrine cells. So amacrine cells are the inhibitory interneurons of the retina that release GABA or glycine, most of them, and or connect through to ganglion cells through gap junctions and or deliver other neuropeptides. So they do a lot of things. And some of them have their somas in the internuclear layer in the middle of the retina where the bipolars are, but a lot of them, especially in a mouse, have their somas in the ganglion cell layer. Therefore, they're called displaced. That technically means they're easy to record because they're sitting on the top when we flatten out the retina. So they're just as easy to record as the ganglion cells. So the difficulty, of course, is that amacrine actually means the word amacrine means axonless. Again, a misnomer because some of them have multiple axons. There's polyaxonal amacrine cells, and some of them have no axons, and some of them have some in between thing of active propagation that's not really an axon. But what they don't have is an axon down the optic nerve. They don't transmit information to the brain. Some of them have axons within the retina. But what that means is that you can't just do a cell attached recording on them. You have to patch them to really understand anything about what they're doing because their spikes are not simple if they have spikes at all. So what was known about these? Not much, less than the ganglion cells for sure. There had not been comprehensive studies like in the ganglion cells. There's this paper from 2007 where just randomly filled cells. This is morphology alone, and they randomly filled a few hundred, and they said kind of looks like there's 10 or so types. But that's about all we really knew. The one type of displaced amacrine cell that is really, really well studied is the starburst amacrine cell. I don't have a pointer, but it's G on that thing on the left, that little one. So I'll talk about starburst a little more in a moment. Those were really well studied, and they're actually a substantial fraction of the displaced amacrine cells. So we wanted to avoid those and not restudy those that everybody had been working on. And we wanted to avoid patching ganglion cells because that was a different project. So we made a transgenic mouse line where we did it by negative labeling. We labeled all the ganglion cells with V-glu2-Cre. We labeled all the starburst amacrine cells with chat-Cre because they're the only cholinergic cells in the retina. And then we crossed that with the AI14 reporter line to make everything expressing Cre turn red. And then we used 2-photon and we hit the cells in the ganglion cell layer that are not red. So that's all we did. And it turns so first we just quantified them. We did all the staining and careful quantification of the fluorescence. And we found that half, like 50% or so of the cells in the ganglion cell layer were not red. So about half the cells are displaced amacrine cells that are not starbursts. So that was our starting point and our method of targeting them. So now I'm going to put up the pillars again, but now we have a fourth pillar, which makes the stool even more stable. We've got function, morphology, and gene expression, but importantly, amacrine cells have very diverse electrical properties too. I mean, we could have done this for ganglion cells, and we actually are doing this for ganglion cells as well. But what I mean by electrical properties is when you inject current into the cell, how it responds, not its whole upstream circuit and how it responds to light, but how it responds simply to current injections. Now, for amacrine cells, that's more informative than it is for ganglion cells. Because for ganglion cells, of course, if you reach threshold, you're going to get some spikes. Now, the spike waveform might vary slightly, the spike rate adaptation might vary slightly, but they're subtle. Amacrine cells either spike or don't, or kind of spike, or have other sorts of weird electrical properties. So there's actually quite a bit you can tell by their electrical properties. And furthermore, we had to patch them anyway to get anything about their light responses. So if you're already patched into a cell in whole cell mode, you might as well inject some current and also get information about the electrical properties. So for all the cells I'm going to show you here, we did function and electrical properties and morphology, and we're just starting the gene expression part. So let me show you what the electrical properties bit looks like. We simply inject current steps, hyperpolarizing and depolarizing, and measure the voltage changes in the cell. And you can extract a ton from this. Those of you who are whole cell electrophysiologists know that you're recording at 50 kilohertz. You've got an incredibly rich data set when you do this kind of thing. So here are some examples of the parameters you can extract from this. The resting membrane potential, of course. But there's this sag potential. If you hyperpolarize enough, you often get a depolarizing step after that. That often comes from an IH current, which is a hyperpolarization activated non-specific cation channel. So that will give you a depolarization that's activated by going to minus 70 or minus 90. You can, of course, measure the resistance of the cell. You just do small hyperpolarizing currents, and you get the slope of the IV curve. You can measure the membrane time constant. You can measure the maximum depolarization overshoot, which gives you a sense of whether the cell is actually spiking or not, and many, many more. So here's my first giant data dump slide. Don't memorize every point on the graph, right? So what's on the x-axis is the 21 types of displaced amacrine cells that we've identified so far from our data set of, at this point, it was 107 cells. I think we have upwards of 200 now. What you'll notice is that there is pretty nice variation in many of these parameters. So every data point is shown. You can see the means and the error bars. And there's actually some really nice clustering. And some of these things vary by quite a bit. Like I'll point you to resting membrane potential. If I did this for ganglion cells, they're all right around minus 60, right? Some of them are minus 55, some of them are minus 62. But there are amacrine cells that are minus 40. There are ones that are minus 60 and pretty tightly clustered around these different values. Same for the sag amplitude. Some have it, some don't. The resistance varies dramatically. Look at the first two columns of the resistance plot. That A1 cell sits right around 250 mega ohms. That SAD cell sits at 500. So these are actually parameters that can be very helpful in distinguishing the types. Of course, we can also measure the light responses. And again, we do so by showing spots at different sizes to the cell, like we did for the ganglion cells. For a spiking amacrine cell, we can measure the spike rate as a function of spot size, and you get curves like this. But for a non-spiking amacrine cell, we simply plot it as the peak voltage change as a function of spot size. So there's a non-spiking amacrine cell. One of the things you can see here is the amount of surround suppression. So this cell has zero surround suppression, which we call it, because it responds just as well to a large spot as it does to a small spot. That's one of the key distinguishing features between them. Some of them have a lot of surround suppression and will start to respond less or more transiently or differently to large spots. Not this one. So another data dump. Here are the wide field amacrine cells. Here are the 11 types of, or 10 types of those that we found. So if you look at this for a moment, you'll see that their responses just look very different, and they really are reproducible, both in current injections and in their light responses, all the way down to some really wild, weird things. Like look at that midnight one down there, which is called midnight literally because Yulia recorded it at midnight and sent me a picture of it. And we haven't come up with a better name yet, but yeah. So that's called the midnight amacrine cell right now. Maybe we'll keep it like that in the publication. But if you look at its light response, look at the look at the light blue and the dark blue curves for a moment. So the light blue is its response to 220 microns. It has that transient kind of response. At 1200 microns, it has like a medium sustained, suddenly goes down, and then has that peak again at the end, right? That looks like just like a mistake or an anomaly of one cell. We've seen it like 15 times. Like that's actually what the thing does. So these are really reproducible and interesting. And most of them are on cells. You see they depolarize or spike to the onset of light, but some of those at the end, like the wide field deep and the bow tie deep, are deep in the inner plexiform layer and are actually off cells. So those are constitutively depolarized at like -40, and then when they see a light step, they actually go down to -60. So in theory, those are probably releasing GABA constitutively in the dark and stop doing so in the light, would be the guess. So here are the medium field types, and again, a great diversity of types. The medium field ones mostly don't spike, which makes sense because spikes are needed for amacrine cells to transmit information over long distances. But if the cell is smaller, it doesn't really need spikes. That doesn't mean it's electrically passive. These things, some of them have spikelets and such, but they don't have like full-blown spikes for the most part. And some of these have more surround suppression too. Of course, we can also measure the morphology, which is additionally really cool in amacrine cells. That's also even more diverse than the ganglion cells. I think you saw a couple of the summary pictures of the morphology of the ganglion cells, which vary in their branch density and their overall size for sure, but not in drastic aspects of their morphology. The ganglion cells tend to be circularly symmetric. They need to collect over a circular receptive field. Amacrine cells look wild. I mean, this top one I used to call the thrown spaghetti, like throw spaghetti at a wall. It kind of looks like that. There's ones that are long and straight. There's ones that look like all different sorts of things. They get very colorful names of spiders and all sorts of stuff because of their very diverse morphology. So we can measure their 2D on phos morphology, and using the same mouse line, I told you about the starburst amacrine cells, which are the only cholinergic cells in the retina. Their dendrites form these chat bands, it's called, that give us a fiducial marker in the inner plexiform layer, the synaptic layers in the middle of the retina, so that we can then use that to computationally flatten the dendritic arbors of the cells, which ends up being very important. So I have a whole software package that, that allows us to do this. So that's a good way to know exactly where each of the cells stratify in the IPL, which turns out to be pretty important for their classification. So here's our summary slide on the morphological diversity of these displaced amacrine cells. Here's the 21 types, and you can look at them for a moment at how interestingly diverse they are. Even more so when you really zoom in. Some of these have varicosities that you can actually see in light microscopy. Some of them don't. Some of them have twisty dendrites with sort of smooth branch angles. Some of them have really sharp branch angles. Some of them are completely long and unbranching, like the NNOS2, which we wrote a separate paper about, or the wide field on spiny sparse. Some of them have spines or not spines, which you can't see at this resolution, but if you really zoom in, you can see the spines on some of them. So there's all sorts of interesting differences in the on-phos morphology, and then the stratification plots are shown below with the dotted lines being those chat bands. And you can see from the individual curves and the averages that these are really tight in their stratification. So for example, look at the wide field on non-spiny sparse and the NNOS2 that's right below it. If you looked at those on FOSS, you'd say they're the same thing, right? They're long unbranching things that go approximately in two different directions. But look how tight the stratification pattern is. One of them is below the chat band and one of them is between the chat bands, and every example falls there. So they're different, and they actually are functionally different as well. Okay, so we haven't done the transcriptome of the amacrine cells yet, but others have without the other pieces. And I wanted to show you a little piece of that because that's where we're headed next in this project. So the Sains lab, again, has been doing this for all the cell types since they helped develop DropSeq in the first place in 2015. And in this 2020 paper, here's the UMAP of tens of thousands. I don't know how many this was, 60,000 or something amacrine cells, both displaced and non-displaced, remember. So now this is all of them. And they say there's 63 clusters. Again, grain of salt. I have no idea if the number's really 63, but it's probably somewhere between 40 and 80. Um, but even more importantly than that paper, this 2023 paper, which is really pretty amazing, this is, in my opinion, one of the best transcriptomic papers that's been done yet, looked at retinal cells across 17 species and found orthologs. They actually tried to match transcriptomic clusters across species. And many of these amacrine cells, they can match quite well from an opossum to a human to a tree shrew to a mouse to a squirrel, a sheep, a cow. Some of them even match to zebrafish and lizards. So this was pretty impressive. And one of the things that was cool about this is that the amacrine cells, even though we don't have as much information about their types yet as we do the ganglion cells, were more well conserved across these species than the ganglion cells were, which my theory on this, I have no idea whether this is right or not, but transcriptomics, of course, doesn't get you everything. It's biased in what it actually gets you. It gets you the genes that are expressed highly. So an ion channel that doesn't turn over, or a gap junction or something that may be very important to the function of the cell, you're not necessarily going to see in transcriptomics. But neuropeptides, things they release, you're more likely to see the enzymes, because you need enzymes that make these things in high volume, and they do turn over. So many amacrine cells, I didn't talk about this much yet, but release a variety of neuropeptides and other things, and maybe the chemical aspects of their function have been conserved over evolution, and that's why you can see the same substance P amacrine cell in a pig that you can in a squirrel that you can in a human. So I think that's neat, but what it means for our project is that when our lab is able to figure out which transcriptomic types these amacrine cells are, we not only have information from mouse, but through this conservation, we may know what type that is in a human or any other animal. So, executive summary on this one, we have 100 to 200 cells currently on our data set. We've clustered them into 21 types. Now, I can't tell you this is 90% done. I don't know exactly how close to done we are, but I can tell you anecdotally that we're not surprised by ones we run into that often anymore. So I'd say, we drop down an electrode, and the vast majority of the time we recognize it as one of these 21 types. So we're probably near the end of this too. I think the answer is probably 20 to 30 or 20 to 5. I don't think there's that many more types than this. We found 11 wide field and 10 medium field types so far. The intrinsic electrical properties differ substantially, and that's been very helpful. So I think they'll be more easily distinguishable than the ganglion cells because we have that fourth pillar. And the morphology and function appear to align, again, very nicely. The patch-seq experiments aren't done yet. Those need to be done to align them with the transcriptomic clusters, but I'm excited to get that started. So we've got our first set of like 8 or 10 samples that we're sending out to the sequencing core now. So we should get there. Okay. Finally, are the cells even the functional units though? So I'm going to tell you about a little bit of published work very briefly about functional compartmentalization in bipolar cells that was published just this year. But this was a former PhD student in the lab who's now moved on as a postdoc at Indiana. And then I'll tell you a little more about the functional compartmentalization in amacrine cells, which is a project that I'm working on myself in the lab, but also with Prina Shroff, who's a really amazingly talented undergraduate student who helps out in the summers with this and did a bunch of recordings last summer. So what if the subcompartments of cells can actually transmit their own different visual information? This idea was shown in this supplemental figure, actually from this paper in 2012 by Asari and Meister. I would argue that this paper had some suggestive data, but was really far from actually proving it. But it was a really interesting hypothesis. And the idea, as you can see in this colored schematic here, is that a single bipolar cell, the excitatory interneurons of the retina that transmit information from the photoreceptors to the ganglion cells, could actually split functional information between different ganglion cells. So no longer is the B itself, the bipolar itself, considered the functional unit, but a piece of it is. Now, these bipolar cells are shown as if they only have like two outputs. In reality, if you actually think the functional subunits might be synapses, we're talking about more on the order of 100 than a couple. So I'll show you in a moment that we actually have a little evidence of this, and more than a little evidence of this in some of the bipolar cells. Then on the amacrine cell side, this is actually not a controversial idea. So on the bipolar side, it's still controversial. On the amacrine cell side, part of this is not controversial because of the famous starburst amacrine cells. So many of you probably know about starburst amacrine cells. They're one of the most famous cells in the central nervous system for understanding computation subcellularly. And that is because they're individual neurites are direction selective, but the soma is not. So what I mean, so first of all, I call them neurites, not dendrites, because they are mixed input-output, right? The actual output synapses are along nearby the input synapses. So this is not a typical integrating neuron where it's collecting a bunch of information at the soma and the axon hillock and then sends something out the axon. That's not how amacrine cells work. So starburst cells have neurites, and this famous work from 2002 and everything. This is a summary picture, but Euler and Detweiler and colleagues showed with one of the first functional calcium imaging experiments that centrifugal motion from the soma outward to the ends of these neurites causes much more calcium influx at the end than motion the other way, inward, centripetal motion from the outside to the inside. So that means that each of these neurites pointing in a different direction is its own computational unit that is direction selective in the direction from the soma outward. And then later work showed that they connect to direction selective ganglion cells along particular orientations, and that's how it works. But importantly, that means if you stick an electrode on the soma of a starburst amacrine cell, it's not direction selective, even though starburst amacrine cells are the key cell that creates direction selectivity in the retina. If you knock them out, there's no more direction selectivity, even though their somas are not. So this is undeniably a subcellular computation. So first a little bit about the bipolar cells. Can the bipolar cell synapses actually be independent computational units? Now let's think about the size of a neuron for a moment. The idea that different pieces of a huge cortical pyramidal neuron could actually be different units isn't that controversial because they're huge. The primary, right, the apical dendrites and the basal dendrites of a pyramidal neuron can get different inputs and they can do different dendritic processing and can have different ion channels, but they're hundreds of microns apart. So this was a big deal when it was discovered, but it's now like understood in biophysical models to be not that surprising because we're talking about hundreds of microns apart, and you can actually have some electrotonic separation here. Here's a retinal bipolar cell to scale. So when I'm talking about computational subunits, I'm talking about different signals there, right? It's a very different size. The actual axon terminals of these things are 15 or 20 microns in diameter. So here's one of these blown up, reconstructed from Rachel Wong's lab with all the synapses identified. It's this tiny little structure has 91 ribbon outputs, which are the output synapses that release glutamate, and 120 inhibitory input synapses. So the potential complexity of this thing is enormous. And even just from an evolutionary standpoint, you might think if this whole thing is one computational unit, an electrotonic structure, why the heck would it bother having 91 outputs and 120 inputs? Wouldn't it be easier and more efficient to stick one big inhibitory synapse there? That would take up less room and energy. So the question is, can they actually be independent computational units? This is very hard to answer at with direct measurements because it's so small. Right? Think about doing calcium imaging experiments here. We tried, we did some, but the calcium diffuses over longer distances than the indicators aren't accurate enough to tell us what's going on submicron kind of scale, nor is the microscope. Even if the microscope is, the indicators are not. And furthermore, functional calcium imaging in the retina, even 2 photon, kind of a bad idea because you're zapping the retina with a laser and you're trying to record light responses. So that's a lot of light. Even 2 photon, you activate the photoreceptors quite a bit. So you can't really get at it directly. But we did our best to get there indirectly. So here are two different ganglion cells. One's called a pixon, one's called an on alpha. They're different in a lot of interesting ways, but one of their striking functional differences is surround suppression, that thing I told you before. So the orange curve is the on alpha and the purple curve is the pixon. And you can see that the on alpha has virtually no surround suppression for large spots, and the pixon has complete surround suppression. It stops firing entirely for large spots. Okay. However, the on alpha and the pixon stratify in exactly the same place in the interplexiform layer. So they are likely to get inputs from the same bipolar cells, but likely and actually get those inputs is two different things, right? Just because the cells are about in the same place doesn't mean they really necessarily get inputs from the same bipolar cells. So to really check this, we did a combined functional EM study with Rachel Wong's lab. So we functionally identified an on alpha and a pixon that were right next to each other, just in a wild type mouse. We just found them by their light responses, filled them with dye, and then laser branded the area so we could figure out where the dendrites of each of the cells were, and then did a dense EM reconstruction for serial block face EM where you could actually see the synapses and the vesicles and count vesicles and figure out what the synapses were between each bipolar cell and each of the ganglion cells. So you can, for the EM specialist, you can see that the synapse identification in this material was excellent. Rachel takes incredible images here. And here's the full reconstruction of all the bipolar cell types that provide input to the pixon and the on alpha. And you can identify the bipolar cell types easily by their morphology, and they form their own mosaics. So here's the distribution. It's not different, right? It's not significantly different between the pixon and the on alpha. So you get the same set of bipolars, and in fact, many individual bipolars in here are synapsing onto both cells. So they get the same input, yet they have different functional outputs. And we showed, so this first figure was the spikes, but in later parts of this paper, we also did this in excitatory inputs, and we showed that it's coming from the excitation. So these two ganglion cells get different functional excitation, even though they're receiving synapses from the very same bipolar cells. Therefore, the conclusion kind of has to be that the bipolar cells have functional subunits, right? So that's kind of where we landed on this, that there actually are functional subunits. We did a lot of modeling in this paper that you can look at for the electrotonic distance. Honestly, at that kind of distance, when we're talking about few microns apart, there's really no way that they have very different voltage signals. But we now think it's probably isolation chemically. We think there's probably local chemical differences that allow the synapses to respond differently at that kind of scale. So ongoing to try to figure out how this all works, but I think it's something to keep in mind throughout the rest of the central nervous system as well, that subcellular processing could really be happening even at small scales. Okay, but then returning to the amacrine cells where we're actively working on this now, how can we look at subcellular signaling in amacrine cells? So there's two different experiments that we've done to start looking at this. So we're patching the somas of these cells so we can inject current at the soma. And while we're injecting current, we also fill the cell with a calcium indicator, a synthetic one, either argon green bapta or cal 520, which actually works better for those of you doing calcium imaging. I would recommend cal 520. And then we can image the neurites at some location with 2 photon. And so we inject current at the soma, and then the question is how much calcium do we get at the different neurites? Now, in a very simple passive cable model, you'd expect something like a decaying exponential, right? You're depolarizing the soma. So all of other things being equal, if you have an even distribution of calcium channels and voltage spread is just passive, then you should see a decay, you should see an exponential decay of calcium following distance from your current injection. Um, we don't really see anything like that. So I'll show you 4 examples of different things. Again, anecdotal examples. We don't have enough population data for me to really tell you what's going on here, but the answer is basically everything is going on. So here's one particular type of amacrine cell where we've done this experiment. So the picture of the cells on the left, the imaging field is shown in the middle there, and the points in the imaging field, the region of interest are colored by their maximum calcium signal to the depolarizing step. And you can see these bright green ones along this segment, they respond with no decay at all, essentially. Like all of these places respond about the same, but then there's hot spots in other locations and other places that don't really respond at all, even though they're also still close to the soma. So regions of neurites in this cell seem to have these like red calcium hotspots, where others have these relatively uniform responses. This cell has kind of some of both things going on. Here's a different looking cell. This is a wide field non-spiny sparse. This is one of these non-branching ones. So here's a piece of its neurites with these soma injections. And here it does look like we have a strong signal near the soma that then decays pretty rapidly. So this one does look like it decays away from the soma, perhaps in a simple exponential way, with a length constant maybe around 20 microns or so. So this is like, I guess, the most normal in a sense. That's sort of a small length constant, but it's what we might expect. Here's another one where this was a cell that was constitutively pretty depolarized. So we were hyperpolarizing it to see decreases in calcium. And this is another one that has hot spots with what looks like kind of a decay around them, but the decay would have to be very small, like maybe a 10 micron kind of decay around the cell, around the hot spots instead of 20. And here's another wild one where we do have a reasonable decay now. So from this darkest red region, you see there is a decay on either side of it, but that red region, that hot spot, is not the spot that's closest to the soma. You actually have a lower calcium and then a hot spot that has a higher calcium response, and then a lower one on either side of it. So maybe the length constant's around 20 microns here as well, but it doesn't even originate at the soma, even though that's where we were injecting the current. So just some anecdotal evidence that amacrine cells are weird, but here's one where we did some light stimulation. I think we found something really interesting. So similar experiment. We record the soma, this time with no current injections, fill the cell again with the calcium indicators. Now we image the calcium in the neurites using the 2 photon, but our stimulus is now light. So we're actually doing UV presentations of light at the photoreceptors while doing the calcium imaging in a different color. And here's one where our light stimulus was drifting gratings at different orientations. So we're trying to test for orientation selectivity. You're going to hear a story here that's very similar to the starburst cell, I think. So this is a cell where I'm showing a polar plot now of its somatic voltage response to the gratings at the different orientations. So the distance from the origin is the response amplitude to each different direction of the moving grating. And this could be argued to be somewhat orientation selective, but not impressively so. It's orientation selective a bit along the obvious direction that the cell sort of goes from northwest to southeast. So it's a little bit orientation selective, but not dramatically so. When we go in and actually look at its neurites, though, you see a very different story. So here's the calcium imaging in this field of its neurites. Now the regions of interest are not colored by the amplitude of their signal, but rather their preferred direction. So let's look at a couple examples here. This blue one up here, look at its signal to 0 versus 90 degrees. So this is incredibly orientation selective to bars that are moving vertically. So they're horizontal bars moving down, which makes sense given that this is a straight neurite going left to right. This one responds pretty equally to 0 and 90 degrees, but is orientation selective around the 45-degree axis because it's a diagonal neurite. This one goes the other way, right? Because it's a vertical neurite. Now it responds great to 90 degrees and not so much to 0. So we're back to what I told you originally about something like a starburst amacrine cell. If you recorded the soma of this cell, you'd have a tough time convincing someone that this is important for orientation selectivity in the retina. But when you look at its neurites, I'd say this is an orientation selective amacrine cell. And we know there's orientation selective ganglion cells. We didn't know where they got their input. I would say this is probably one of the places they're getting their input. We don't know. We need to do paired recordings, do the connectomics, and figure it all out. But this argues that amacrine cells can do interesting feature selectivity in subcellular compartments, not just the starburst cells, but other ones can too. So very ongoing, very early in our work on the functional subunits, but executive summary here, are the cells even the functional units when we're talking about amacrine cells? No, they're really not. Pieces of them are. In many cases, amacrine cell neurites or even nearby bipolar synapses tens of microns or 5 microns away can potentially transmit different visual information. So this makes the computational complexity of retinal circuits enormous. And I would argue that this is probably true in most of the rest of the central nervous system too. People just don't necessarily want to think about it yet. It's very important in the retina to pack as much computational power as you can into as little space because light has to go through it. So it has to be a relatively transparent tissue. So space is certainly a huge constraint in the retina, but it's not like space isn't a constraint in other parts of the brain. And this kind of thing is probably happening elsewhere as well. So to summarize, we have a long way to go. Retina is not a video camera, but retinal neuroscience is in a position to lead in understanding and really grappling with that complexity. So I want to thank everyone who did the work. The people highlighted in red are some of the key people who led this research. David, who's an alumni from the lab and is now a postdoc in Indiana. Yulia and Zach, who are still postdocs in the lab doing all sorts of other exciting things in addition to this. They each have several of their own projects and things they're working on together that are really cool. My funding sources, all my collaborators, including Tudor, who's listed first, my most valued collaborator right now, who just wrote a grant together on this metabolism stuff. And thank you all for your attention. I'm happy to take some questions.
Greg, this was fantastic. And besides the lesson in neuroanatomy, physiology, and molecular retina classification, these last 2 bits were really interesting and thought-provoking. I have 100 questions, of course, but I would first ask people that are not retina physiologists ask their questions and see what interesting things they found in your presentation. So,
Great.
do we have any takers?
Who is the first?
Hello?
Hello.
Okay, Vladimir, go ahead. You are also kind of a retina person, but let's say you can ask a question.
I'm not physiologist, so anyways, Yeah. So, Greg, thank you very much for sharing new interesting data, lots of new stuff which I didn't know, especially with this topic of compartmentalization. It was known that starburst amacrine cells have this compartmentalization, but not many other cells in retina I heard about. So, what would be your hypothesis? How this compartmentalization is established during development? Is it just simply by the lamination pattern they connect to every amacrine and ganglion cell, whichever is on some specific sublamina, or there might be some specific cell adhesion
Oh yeah. Yeah.
cues for the
Right.
different parts of
Yeah.
the molecule.
Absolutely. Right. Okay. Yeah, I see where you're going with. I'm not a developmental biologist, but like this is a huge and super exciting question for developmental biologists and people who actually are doing this kind of molecular stuff. So it's not even really known how that works. It's not known really at all how that works for the famous circuit of the starburst amacrine cells and the ganglion cells. It's known that it does work like that, that the one that the starburst, the point up, provide inhibition to the ganglion cells, the preferred downward motion. Like that
I
definitely is
think you're
known.
talking about some molecules, but I'm
Right,
not
but the
sure.
molecules, yeah,
Yeah.
the molecules, there's very little known about how that actually works, and it has to be subcellular, right? Like,
Mhm.
which is kind of wild. Like, that's why it's been tough to figure out because it's not like you can do the transcriptome of a starburst and the ganglion cell and ever
Yeah,
know
yeah,
that
yeah, yeah.
because it's
Yes.
right, it's a
But,
subcellular thing. But that's probably true in many of these cases as well. And with the bipolar cells, one of the things I'll say is that I showed you that it's got a lot of output synapses, those ribbons. You may know that those are actually dyad synapses. It's not just a bipolar cell ribbon connected to a ganglion cell. Like
yeah.
90% of the time, there's an amacrine cell sitting there too. And those amacrine cells that are sitting right at that synapse, like nanometers away, actually get there first in development. So it's possible that the amacrine cells are instructing the particular connectivity between particular
The
synapses.
same what they say about starburst amacrines, yeah, like it's
Yeah.
establishment. Yeah. Okay. Yeah. Actually, what I noticed for your specific experiment about M4 and M5, that it might be also layer specific. Yeah. Whatever is in the layer, then bipolar is simply, attaching to those guys. Maybe some micro, distances.
Oh, micro layers. Yeah, so they're basically in the same layer. They're in very overlapping layers, but there's a tiny upward shift of one of those cells relative to the other, but very small. And we looked at that. We saw if that electrotonic distance was enough. It's really not. Like basically in the same layer, you can have synapses onto a pixon and an on alpha
Mhm.
5 microns away from each other. So
Okay, so it's
like
actually different.
uh,
Okay, cool to know.
it's really a small distance.
Yeah.
It's too small to be anything electrotonic. It's got to be kind of chemical isolation, I think.
Okay. Yeah. Cool. And another one is rather provocative question, not really attached to the topics you mentioned, but on the one of the first slides where you had the targets of innervation, there are some new or how would you describe them areas in the hypothalamus which are
Oh yeah.
which are mentioned by Gray. Is it
Oh yeah, there's all sorts of places. Okay, so I'm not the person who originally said that these are all the areas. This is from a big study for the Allen Institute. They came up with the number 59 by injecting hundreds of mice with GFP and sectioning every brain the way the Allen
Mhm,
Institute does, and looks at the places where they saw any GFP at all.
mhm.
And yes, that included medial amygdala, which is involved in identification of mates and social behavior. It involved lateral hypothalamus, which is in feeding behavior. I mean, and mood, like all sorts of areas that are involved in very interesting behaviors. That's actually why we got into social behavior. So we found some work that's going to be published soon on social behavior. There's a ton to be done here. I mean, this is the next decade of work in my lab, is to figure out where all the ganglion cells go to all these different areas and then try to figure out how they're involved in behavior. It's going to be a hard task. Many of these are sparse projections and they're interconnected brain regions and they're multi-sensory. Like, it's not just going to be as easy as the beautiful example of the ones that go suprachiasmatic nucleus control the circadian clock. That's really great.
So they could
That's
be
awesome.
discussed, yeah.
But
Okay.
it's probably not that simple for most of these other areas, but we're trying to figure that out.
So,
Yeah,
Vladimir,
thank
now
you.
given
Yeah,
that
that's it.
you
Yeah,
have
that's
talked
it.
and will talk in the future, hopefully with
That's
Greg,
it. Yeah.
even from Novosibirsk or even from some other place, let's
Magadan.
leave some other people ask questions. So Anna
Yes.
Maria, I think, was first raising her hand.
Okay, so Ana Maria Zagran from Carol Davila University in Bucharest. Thank you, Greg, for the fantastic presentation. I think that all of us here researching in neuroscience can fall in love for retina research, because actually it's amazing what it's a hot topic still, and it will remain, and it's a long time to discover how the retina process all information that can get. And regarding this, of course, there are many questions, but just to when we teach students about synapses and connections in between cells, we also mention the electrical synapses, the gap junctions, and it is known that at the level of retina and in the retina circuits, these electrical synapses are present. And my question for short would be, how the homo and heterocellular connections through electrical synapses influences the retina circuits, and how this, if it's known, would impact the retinal circuits during neurodevelopment, because it is known that early in the immature brain, there are more electrical synapses that modulate and sculpture the circuits. Okay, you understood that.
Oh yes,
Okay,
absolutely. How did you know? It was one of my very favorite topics. I can't believe I talked for an hour and didn't say anything about gap junctions. I've written many papers about gap junctions in the retina. It's one of my favorite topics. So I'll hit the development piece first because I know less about it, and then I'll talk more about the other parts. So this is not my work, but it's definitely known that gap junctions between ganglion cells are more extensive. They do exist in the adult retina for sure, but they are more extensive in development, and they're actually they're required for retinal waves at a particular time point. The early waves, the very first stage of retinal waves, rely on gap junctions between the ganglion cells, because actually at that stage of development, the ganglion cells aren't connected to photoreceptors anymore yet. So there's no light sensitivity from the photoreceptors. The light sensitivity comes from the intrinsically photosensitive ganglion cells and then spreads through gap junctions throughout the retina. So in development, that actually is important for establishing the retinotopy of the synapses between the retina and the superior colliculus. And it's all dependent on gap junctions and intrinsically photosensitive ganglion cells. So that's the piece that I know in development, but there's many more interesting pieces along that line in development. In the adult retina, there's tons of cool things that the gap junctions do. Gap junctions in every one of those 5 cell classes of the retina has gap junctions: photoreceptors, amacrine cells, ganglion cells. There's gap junctions between amacrine cells and ganglion cells. There's all sorts of things. But let me tell you 2 tiny little vignettes that we've worked on in my lab. We discovered the first heterotypic ganglion cell gap junction, and it's actually, believe it or not, between an on and an off ganglion cell. This is another one you wouldn't hear in the textbooks, right? So we have an on ganglion cell and an off ganglion cell that stratify respectively in the on and the off layers. You actually record them, and they both respond to on-off. And it's because they're gap junction to each other. If you eliminate the gap junctions, then they become strictly on and strictly off. So
yeah, it's
actually,
amazing. Yeah.
and they're actually spatially offset from each other. So what you get is like what's been described in V1. You get like a stripe receptive field where you have the on and then the off is actually 20 microns below it. This was the paper we wrote in Nature Neuroscience in 2021, I think. So yeah, you actually get like what looks like a V1 receptive field in the retina because of this heterotypic gap junction. And homotypic gap junctions, there are a ton of. Those nNOS amacrine cells are a great example of that. So that's one of these really long straight ones. Right? That was one of the types I showed that had those really long straight neurites. So you'd think it would be orientation selective, or at least pieces of it would be. It's not at all because its gap junctions are so strong that more of the signal comes from the cells around it than from the actual neurite itself. You fill one of those cells with a dye, you get 50 labeled. It fills the entire retina. So those ones have really extreme gap junctions.
Okay, and then we might say that the computational units are even more complicated, and also through these gap junctions, there is a huge communication in between different computational units.
Yes. Yes.
Something
exactly.
that it will be, and that's why no camera can compete with an eye with retina
right.
because there is the features and the variety of features that can be perceived
yes,
it's
incredibly flexible and it's huge. And actually the ones between the bipolar cells, like another example of that is the ones between the bipolar cells give it motion sensitivity. So like as a bar is moving across the retina, some of the excitation gets kind of pushed forward through electrical synapses from one bipolar cell to another. So that means that then when it's like a traveling wave, when the bar gets to the next, it's already primed. So like it's actually inherently motion selective because of that.
Okay, and you mentioned about during neurodevelopment, and I will not insist on this, but it is known that neurons and cells during development needs excitation, and then maybe through these electrical synapses, there is an excitation in the circuit that without visual perception actually.
Exactly. No, that's definitely
kind
true.
of maintaining the circuits and paving the way for future connections. Okay, thank you very much, and I hope we will see each other next year.
That'll be great. I'd love to come out there.
Okay, I am waiting to see you. So thank you very much again, and all the best. Success.
Tudor? The other Tudor?
Yeah, hi. Hi, Greg. Thank you for the great. So, can you comment for us briefly what do you think about direction selectivity in the human or primate retina and how this compares to the rodent retina? How your discoveries maybe are paving a way for discovering direction selectivity in the primate retina. I know that for a long time it was considered that direction selectivity is not present in the primate retina, but there are new works. I think.
Oh yeah, it is. It totally is.
That there is something.
It absolutely is. Yeah. That Nature paper that came out of Teresa Pusatari's lab, I think was really definitive in that. I mean, 3 papers came out at the same time, similar time last year that showed things about that. I really think it's just sampling. So I've worked a little bit in the primate retina too. And there's this kind of myth out there that the primate retina isn't as complex as the mouse and doesn't have as many cell types. And I think that's just mostly wrong. I think it's just sampling bias. Like it's got an enormous number of midget and parasol cells because we have a fovea and they're incredibly dense and they're 10 deep. So yes, when you actually numerically count, 75% of the cells are either midget or parasol, which makes it very tough to find all the other sparse types. But you go to the peripheral retina, they're absolutely there. There's also just way fewer labs working on non-human primate retina, so we don't have as much information about all the cell types, but there's been more work now. And I think in the next couple of years, we're going to arrive at a number that's more than 30 types of ganglion cells in the non-human primate retina as well, which are transcriptomically and morphologically almost perfectly aligned to what's in the human retina. So do we have 30 or 40 cell types? Yeah. The actual distribution of numbers is probably different, but we have the same number. And direction selective ones have absolutely been found. The ones that Teresa found were actually the on-DS cells. And one of the coolest things about that story is that, from my perspective at least, biased, is that the way she marked them in the first place to look for them in the primate is by using our transcriptomic data to find the homologous gene in a primate and to stain for it. So like we figured out which transcriptomic cluster was the on-DS cells in a mouse. They found the homologous one from the primate transcriptome, figured out what the marker gene was, and lo and behold, that's the same one that's the on-DS cell in a primate. So yes, they definitely have direction selectivity. They definitely have starburst amacrine cells. And yeah, it's probably doing similar things and going to similar places in the brain.
Okay, great. Thanks.
Okay, any other comments, questions? Yes, Professor Frontam?
Can you comment? Your presentation was very, very interesting. But I would be interested if you can comment something about myopia.
Yes. Yeah. Oh, I could talk for a long time about myopia. So, yeah, so again, not my work, but I find it a really super fascinating question because all myopia is your eye growing to developmental myopia in children is your eye growing too big so that the optics make the image focused in the middle of the eyeball instead of on the retina, right? That's physically all it is. So it's a problem of eye growth. It grows a little too long. So what regulates eye growth? It has to be regulated super tight. Obviously, when you're a kid, your eye is small, and when you're an adult, your eye gets bigger, and it has to stop growing at exactly the right size to be optically fixed correctly. So that process of deciding how much to grow your eye is internally regulated. And this is experiments back to the 80s and 90s done in chickens and mice and other rabbits and other animals, which shows that you can put a lens on half the eye and that half the eyeball grows a different length. And this whole thing works with the optic nerve cut. It has nothing to do with feedback from the brain or anything else. So how in focus the image is in your eye determines some growth factor that changes how much the eye grows, which I think is just a really cool neuroscience question, right? So what cell is the focus detector? How does it know positive from negative defocus? And then molecularly, what's the growth factor? So we wrote a paper a few years ago about a cell that's a potential ganglion cell that might be the focus detector because it's very sensitive to slight defocus. And we now published a more recent paper that shows that it actually might have color sensitivity that helps us know the direction of defocus. Because any of us who use a microscope know that when you go out of focus, you don't know if you're out of focus up or down before you move the knob, right? You see if it gets better or not. The retina knows immediately. It knows negative from positive defocus. And one theory for how that could happen is chromatic aberration, because chromatic aberration has a direction. Shorter wavelengths are closer and longer wavelengths are further. So actually the color sensitivity of this cell lines up with what you'd expect for a cell that could do the chromatic aberration thing. And then my crazy theory about the molecular mechanism is that some ganglion cell or amacrine cell releases a molecule that travels to the outer retina and actually interacts with ion channels in the retinal pigment epithelium, because the retinal pigment epithelium has these tight junctions. And if you change the permeability of the retinal pigment epithelium, the RPE, slightly, there's an osmotic gradient across it, and it makes the whole back of the eye thicker or thinner. And some recent work has shown that that can happen within 30 minutes of focusing in different locations or reading black on white text versus white on black text in humans. So I think what the whole thing is basically you have some cell that detects the direction of defocus, it releases a growth factor, it makes the back of the eye transiently get thinner or thicker and push itself, and then long term that creates remodeling of the whole sclera in the eye. So I think operationally what that means for kids with myopia is that if we found a way to interact with the focus detector cell in the first place, like pharmacologically, and I have some ideas there, that would be good. But also, if we designed contact lenses and glasses to correct for the defocus chromatically, in addition to just like optically do the chromatic aberration correctly in the peripheral retina where this is, you could actually get myopia under control optically. So I think but yeah, I need collaborators to really do this right in mouse, because people do the measurements in mouse, but it's hard. The myopia is small, it's only a few microns, and you have to like stick a lens on a mouse surgically and got too many other things going on to do that right now. But yeah, I think there's some super exciting things that need to be done in that field.
Thank you.
Okay, just a comment on that. I believe there is one of the other OPNs is implicated, OPN3 or OPN5. There is marks some other ganglion cell populations. Yep. And there are experiments claiming that modulating OPN, I forget which population that was, can
yeah, it's a big mess of a population. Yeah, we were on that paper too. Yeah, it's F minis and a lot of other stuff. But yeah, maybe OPN5. I think it's developmental, but I don't know. Yeah. I don't know.
And yeah, that is amazing. So yeah, these were all interesting discussions. And Greg, I'll catch you some other time for my own curiosities, and then we can talk later. And thank you very much for this amazing talk.
Yeah, it was wonderful.
Hopefully, again, see you end of May in Brasov. So make a tentative schedule arrangement there to see whether you're available at that time.
Sounds great.
Hopefully, there's going to be the beginning of a vision meeting that I planned for a while that I want to organize there. And I discussed with a few other people, and we realized that while there is the retina circuits meeting, there is not a circuits meeting going upwards, like superior colliculus, LGN, cortex.
Yeah, and I have some good colleagues working on some of that stuff now. SC, we have a guy who we just hired who's doing that. So.
And there is a disconnect in the world of people thinking about the cortex and then people, delivering the message from the retina.
Yeah.
Okay, so I suppose this is, I'm going to stop the recording now, and also thank Greg again for the participation, and then all the guests that were able to attend. And
thanks so much. It was wonderful. Great questions. A lot of fun. Thanks very much, Tutor.
Take care.